Hello everybody and welcome to your video lecture on plant nutrition. Now the way you should be using these videos is to consolidate your understanding after the inquiry. Probably best not to do them before the inquiry because it might just give a little bit too much away. So the videos are really going to summarize what you've done in class and kind of complete that and hopefully fill any gaps that we might still have. Okay, so our definition for photosynthesis, what do we need to know about it? So it's the fundamental process by which plants manufacture carbohydrates with carbon dioxide and water using energy from light. Now, I don't want to just read that to you. Let's just make sure we've unpacked it and we can understand it. So the process by which plants manufacture or make carbohydrates. So our carbohydrates that we're talking about, of course, are glucose or sucrose or even indeed starch. And they're making it from carbon dioxide and water using the light energy. Okay, so we're going to be looking at how that actually happens in this unit. So the fundamental process of trapping sunlight energy. Hopefully you'll remember from the actual unit that palisade cells in plants, which are the cells which are on the top layer of leaves, contain small organelles called chloroplasts, and these are responsible for trapping light. And so the way they do that is they trap the light coming in, so light energy goes into them, okay, and they convert that light energy into chemical energy in the form of carbohydrate. So what really generally happens then is they usually make the stuff into glucose. That's the first sugar that they make. It's often then changed into sucrose for effective transport. And then it's changed into starch for storage. Now, the reason for that, of course, if you remember that starch is insoluble. And so when we store it in cells, it doesn't cause any changes in the water potential of those cells. So it's not drawing in water or causing water to go anywhere. And of course, oxygen is released as a waste product or used in the plant for respiration. So when we photosynthesize, carbon dioxide and CO2 are used, so CO2 and water are used, and oxygen is made as a waste product. So our equations that we need to learn, of course, we've got the word equation, carbon dioxide and water. We have a chemical reaction which is signified by our arrow, okay, and what's released then is glucose and oxygen. The glucose then can be used for respiration or stored as starch, or like we said before, transferred into sucrose, a different type of sugar for transport around the plant. As a balanced simple equation or chemical equation, it's 6 molecules of CO2 plus 6 molecules of H2O, gives us 1 molecule of glucose, which is C6H12O6, and 6 molecules of oxygen or O2. Okay. So one of the initial experiments that we did is we tested leaves for starch. And you can remember the way we did this. The first thing we did is we boiled the leaf in water, which was to make the leaves permeable. We then boiled the leaf in ethanol, which was to dissolve the chlorophyll from the cells, making sure, of course, that we never put it anywhere near a flame. We then rinsed the leaf in water, which is to soften it back up again. And then we put the leaf onto our tile and we bathed it in starch and looked for a colouring. Now remember, if it was a black colour, that meant starch was present, and if it was a brown colour, it meant that starch was not present. And we use that as a way to determine whether or not photosynthesis had occurred. So if leaves had starch, which obviously are a store of glucose, we could say that photosynthesis had been happening in that area. And the 3 experiments this was relevant to is when we were looking to see if chlorophyll was needed for photosynthesis. The way we did that is we used a variegated leaf, which meant it had green areas and it had clear areas or white areas where they had no chlorophyll. And the result we got was that the green areas would turn black and the white areas turned brown. So the black areas had starch present, showing that photosynthesis hadn't been occurring, and the white areas were a negative test for starch. Second test where this was relevant is we did an investigation, or we looked at planning an investigation to test whether CO2 was needed for photosynthesis. And the way we did this, obviously for this test and the one after it, we need to de-starch the plant first by keeping it in the dark for 24 hours. We can then attach flasks or plastic bags, each one with a carbon dioxide absorber like lime water or potassium hydroxide in it. The other is a control with water. Around the mouth, if we're using a flask, then we need to put something that's going to stop CO2 getting in. So in this case, there is cotton wool soaked with lime water to absorb any CO2 from the atmosphere, and the same on this, and we don't need to do the same with this one over here. And then, of course, after let it photosynthesize for 24 hours in the sun, and then repeat the experiment above. So repeat this experiment again and test them for starch. And what we'd hopefully find is that this one would test positive for starch because it would hopefully photosynthesize, and this one would test negative for starch. To see if light is needed for photosynthesis, we do the following. Again, we destarch the plant by leaving it in the dark for 24 hours. Then we attach black paper with paper clips, leave it in the light for 24 hours, and then test one of the leaves. And hopefully, once we've tested the leaves for starch, we would see that this area here would turn a black color, indicating that photosynthesis has been happening. This area would be brown, indicating no starch and no photosynthesis. The online experiment that we did was to see how light intensity affects the rate of photosynthesis. Now, we all managed to find that as we increase the light intensity, the rate of photosynthesis increases. And the way we did this was by using a plant called Elodea cannabis, which is Canadian pondweed, and we did it by calculating measuring the number of bubbles per minute. Another way we could have done this was by using displacement. We could have used an inverted tube over the top and measured the amount of gas that we collect in 1 minute. We might have also measured the amount of carbon dioxide used or the actual amount of oxygen produced using a gas sensor. So there are different ways to measure it. Now, this graph here shows us what we would typically expect. So you can see here as light intensity is increasing, okay, the rate of photosynthesis increases. Now you see that pattern doesn't actually last forever. We get to this position here and it starts to plateau. Well, the rate is at its maximum rate. Now there's a special reason for this, and it's because another limiting factor has come into play. So if you think about the factors that affect photosynthesis, okay, it could be that there's not enough CO2 to drive the process, and so now we haven't got enough CO2. This is where CO2 has become limiting. It might be that all the chloroplasts within the leaves of the Elodea are photosynthesizing at their maximum rate, and so it can't go any faster, and so the number of chloroplasts might be a limiting factor. Water could become a limiting factor, and of course, this is light intensity. That's probably the main ones we've discussed. So please bear that in mind. If your graph showed that plateau, that's absolutely fine. Right, we're going to stop video 1 there, and I will see you later for video 2.